to extend the classroom learning and also to inculcate scientific temperament in students, school students particularly. As a part of Jigyasa ATL, this webinar series was launched, which had 15 webinars during June 15 to 22nd September 2020. And these webinars attracted a large number of students across the country. So the overall webinar series had a massive reach through social media platforms. And today we are restarting this webinar series, and it is positioned as just Swati mentioned, Demystify Science series. Yeah. So this is a part of Azadi Ka Amrit Mahotsav initiative of CSIR and NITI Aayog. And as name suggested, the webinar series is expected to demystify various science myths and provide a scientific perspective to all the children and enabling them to understand nature better. So today, for the first talk in this webinar series, we are honored and privileged to have with us Dr. Shekhar Mande, DG CSIR and Secretary DSIR. Welcome, sir. Welcome. Before we start, let me give you a brief introduction of Dr. Mande. Dr. Shekhar Mande is an Indian structural and computational biologist. He did his MSc in physics from University of Nagpur. He holds Doctor of Philosophy in Molecular Biophysics from Indian Institute of Science, Bangalore. Following his PhD, he joined Professor Wim G.J. Hall as a postdoctoral fellow in University of Groningen in Netherlands. Since 2001, he was a senior staff scientist at Center for DNA Fingerprinting and Diagnostics. Between September 11 to September 18, he's Pune, India. And Dr. Mande is also a recipient of many of awards and honors, such as Bharat Asmita Tantra Vigyan Puraskar, Shanti Swarup Bhatnagar prizes for biological sciences, and many more. So we welcome you, sir, and over to you.
Thank you, Shubda. I hope I am heard well.
Yes, sir.
Thank you, Shubda. Thank you, Swati. Thank you, Geeta. I am honored that Atal Innovation Mission and CSIR Jigyasa program have started this Demystify Sciences series. And I believe I am the first speaker in this particular series. So I'm deeply honored by this particular gesture from both AIM and CSIR Jigyasa program. As both Swati and Shubda told you, that the objective of Atal Tinkering Labs and that of CSIR Jigyasa program are to reach out to students in very large numbers and to tell them how exciting our science is. There are questions about nature all around us that we need to keep probing, and answers to many of these questions are not known. And that's precisely what the scientists like us are doing in our own labs, trying to find out answers to some of the most difficult and deep questions. One of the questions that excites many people like me, are how do we distinguish living forms from non-living forms? We know that there are many living forms around us, plants, animals, insects, we human beings. As we know, we are all living, and we know living forms have particular characteristics. With the non-living forms, the glasses that I'm wearing, the clothes that I'm wearing, stones, water, they're all non-living and are distinctly different from the living forms. Why is that? What is the difference between the two? That is a very deep question, and let us try to see whether we understand any of this in the next 35-40 minutes or so. So welcome to this series. So the living form and non-living forms, I already told you the differences. For example, stones are non-living, grass, what we call living, water on a gas being boiled in a pot, non-living. Although we do see actually vapors coming out of the water and the flame is moving, but nonetheless, we know that this is a non-living form. While the plant here, which is a cactus, although it doesn't move, it's a living form. The tortoise that you see here, turtle, is living, while a dead wood is not living. So there are the kind of differences that we have. And the question we ask is, what is common between all the living forms that we see here, which is not there in the non-living form? And answers could be many. It's unlikely to have one single answer to this. But some of the answers, if you ask different people, they will say living things are made up of cells. Indeed, they are. Living things obtain and use energy. Indeed, of course. Living things have the ability, as we see here, is whatever energy around us to assimilate that energy and use it for their own growth and development. Living things grow and develop. Living things reproduce. Living things are so good that they make copies of themselves. We make copies of human beings, right? Our children are copies of ourselves. We ourselves are a copy of our parents. And living things adapt to their environment. Living things have remarkable ability to adapt to the environment. So these are some of the answers, and not all the answers that characterize living things from non-living things. But one could actually keep coming up with some answers and then say that these are the differences between the two kinds of forms. How did this come about? When did the first living being come on the planet? And how do we find so much diversity in the living things on the planet today? That's a very, very deep philosophical question even today. Let us look at, for example, the scale changes in the living form. Scale changes in the sense, we are going to talk in terms of meters, centimeters, micrometers and all. And as all of you know, what you see here is blue whale is about 10 meters long, while humans are 1 to 2 meters long, centipede is about 0.2 meters, a small spider is about 0.02 meter, small ant is about 0.002 meter, red blood cell is 20 microns, the bacterium is 2 microns, the virus is 100 nanometers, the DNA is about 2.5 nanometers, and atom is about 2 nanometers. So what you see here, if we reduce ourselves 1/10, 1/10, 1/10, 1/10 like that, from all the ways, the largest living form that you see here is the whale, and the smallest entity in the universe that we know of is an atom. We have literally about 7 to 8 steps from the largest living organism to the smallest entity in the universe we can reach there. Is it literally about, we just traverse about 7, 8 or 9 steps. Every time reducing the dimension by about 10, we came there very easily. And that's remarkable. What we need to do is actually ask a question now that we know that we are all surrounded by atoms and molecules. And what are the kind of molecules that they go on forming the living systems which are not there, or if they are there in the non-living systems, but they're not used for the same purpose like what we saw, reproduction, growth, development, and so on and so forth. There's a remarkable unity of diversity in terms of molecules that make up all life forms. It's absolutely fascinating. In fact, all the living systems are made up of carbon, hydrogen, nitrogen, oxygen, sulfur, phosphor, and a few other atoms like manganese, iron, and so on. But these 7 that I have written here, the 6 I have written here, make up more than 99% of cellular mass of any living system, whether it be a bacterium, whether it be a plant, whether it be an animal, whether it be an insect, whether it be human being, 99% mass of our cells is made up by these the things alone. And in fact, if you go to the molecular structure, one of the dominant molecules of life is water. It makes up 70% of our cellular weight. But there are also other molecules such as inorganic ions, sugars and precursors, and so on and so forth. They also make living systems. So if you look at the diversity of living forms, there's a common thread amongst all of them, that they are all made up of very similar molecules, what we see here on this slide. And that's remarkable. As I said, water makes up 70% of cellular mass, and water therefore can be considered as one of the most important molecules of life. Why is that? Let us quickly look at the structure of water. As all of you know, water is made up of 2 hydrogens covalently bonded to an oxygen. Now, oxygen also has 2 lone pairs of electrons on the top. a lone pair here and a lone pair here. One would have anticipated that this would form a tetrahedral structure. And indeed, the two lone pairs and the two hydrogens almost form a tetrahedral structure. But a small difference. And the difference is that the angle between hydrogen, oxygen, hydrogen bonds is 104 degrees, which is contracted by about 5 degrees. In a classical tetrahedron, the hydrogens would have been slightly apart from each other at roughly about 109 degrees. And therefore, the hydrogens are closer, and that gives water some very, very fascinating properties. We all know hydrogen is the smallest atom that we know, and if oxygen attracts the electron of hydrogen towards itself a little bit, both the hydrogens will be slightly positive in charge, and the oxygen would be slightly negative in charge. And indeed, a water molecule has slight positive charge on this surface and a slight negative charge on this surface. And that makes water very polar. And that gives water many of its unique characteristics which sustains life. And that's why water is a very, very, very important molecule of life. Let us look at only one property of water, how life forms use water for their own advantage. One property, we are not going through all the properties, there are many other properties of water which are important for sustaining life. But one property is that water has a very high heat of vaporization. That is, the amount of heat needed to turn a given amount of liquid water into water vapor is very high. What does it mean? It means that when we do a very rigorous exercise, our body generates a lot of heat, and that heat is not good for our body. So what do? We perspire. As what you see here on the figure, Roger Federer, after playing a very rigorous game of tennis, is perspiring or is sweating a lot. And the sweat has a very natural cooling mechanism because the sweat evaporates on our skin. And the moment the sweat starts evaporating on our skin, it releases a lot of heat in the air. What has water done here? Because of its high heat of vaporization, water has offered us a natural cooling mechanism of our body. Any point of time, our body has generated some heat. Immediately, water offers a natural cooling mechanism. And see how beautiful this example is. That the molecule of life, water, helps us in avoiding excessive heat generation in our body, or rather cools down our body when there is excessive heat that is generated in our body. But there are many other molecules that make our life interesting. I told you some time ago that many life forms, or rather all life forms, are made up of very similar kinds of molecules. And these kind of molecules actually were discovered by a very famous experiment in 1953 by two gentlemen, Mueller and Urey. And 1953, incidentally, since I have written here on the slide, was a very interesting, fascinating year. Just for the benefit of the school children here and the school teachers here, 1953 was also when the structure of DNA was discovered by Watson and Crick, with of course help from Rosalind Franklin and Maurice Wilkins. 1953 was also a year when Everest was conquered for the first time. So Mueller and Urey, the two gentlemen here, they did a very fascinating experiment. They asked a question to themselves: how did life come upon on this planet? And to answer that question, they took water in a flask and they simulated atmosphere of that time, which was methane rich, and they started boiling water, and then they passed some electric sparks through this because there must have been a lot of lightning and all in the atmosphere. And when the lightning and all started here, they collected some of the molecules into this flask here. Whatever molecules that were being generated here, they were collected in a trap here. And lo and behold, what would Mueller and Urey find? They found traces of the molecules that make up life today. The molecules that make up life for all the life forms today, they were found there. So the molecules which make up life today are molecules such as amino acids, nucleotides, lipids, sugars, and so on and so forth. And let us briefly look at what these molecules do. So what Mueller and Urey hit upon was a fantastic finding that life must have started with these molecules at that time, and at that particular point of time, the molecules found a way of assembling upon themselves, what we call self-assemblies. And once the self-assemblies started, the early life forms discovered a way of replicating themselves, making copies of themselves. And that's how we believe the life would have formed at that particular point of time. Let us look at this building blocks of life very briefly before we go further. As I said, all life forms that we observe today on the planet are made up of cells. Bacteria, plants, fungi, they're all made up of cells. Our human body is made up of cells. And the cells, because cells have an inside and cells have an outside. And the inside has to be protected. As I said, inside the cells, a large fraction of it is water, but there are many other molecules as we saw on the previous slide. So the cells have one important aspect to do, is that water formed inside the cell should not be exchanged easily to the outside of the cell. And that job is being done by the cell membrane. What you see here, this particular is cell membrane that prevents any of the things which are inside the cell to be exchanged freely to be outside the cell. And that's a very important point, because inside the cell, many different kinds of things are happening. The cells are trying to replicate themselves. That's happening inside the cell. Cells are metabolizing inside. That's happening within the cells. Cells are using energy inside. They are doing it within. And none of this should be exchanged freely with outside. If at all there is any exchange, for example, uptake of nutrients, or for example, uptake of oxygen or something, that must be regulated. And the regulators indeed are offered by some of the regulators which traverse the cell membranes, so that things from outside can be brought in and things from inside can be taken out, but in a very controlled, regulated manner and not in a free manner. And the cell membranes are made up of molecules, what we call as lipids. What you see here are the lipid bilayers. Lipid bilayers are protective of insides of the cells. Lipid bilayers offer a mechanism to transport in and out of certain things from the cells. They also allow cellular recognition. How do cells store energy and transform information from generation to generation? There's another question. The major molecules of energy, and as many of you would have heard in your syllabus, the most common molecule of energy is, of course, ATP, correct? Adenosine triphosphate. But adenosine triphosphate itself has certain sugars in it. And sugar, therefore, turns out to be one of the major sources of energy for all life forms. Whether it is 6-membered sugar, glucose or fructose, or whether it be 5-membered sugars, what is called as deoxyribose or ribose, they play very important in the sustenance of life forms. Energy derivation is from glucose and fructose like molecules, whereas information transfer from generation to generation is by using molecules such as deoxyribose and ribose. And let us look at how information is transferred from generation to generation. Have we not asked a question to ourselves that the babies of tigers are tigers themselves, and the babies of monkeys are monkeys themselves? Have any one of you observed that a baby of a monkey is a tiger and a baby of a tiger is a monkey? No, we have not heard that. So that means that there's a information, very specific information transfer. The tigers can give birth to only tigers and monkeys can give birth only to monkeys. We human beings also give birth only to human beings. Going a step further, you will realize that the similarity between different generations does not end there. The similarity is much more deeper. Let us look at a figure of two very well-known personalities, father and son. And I don't need to tell you who this father and son are. And look at their facial features. Aren't they similar? They're very, very similar. So all the children have facial similarities, behavioral similarities with their parents. And that what we call is an information transfer from one generation to another generation. And in fact, if the babies did not have resemblance to their parents, there would be something very serious, to some doubts that would be raised. And let us not go into those. But nonetheless, information is transferred from one generation to another in a very, very effective manner. And how does that happen at the molecular level? Answer is very simple. At the molecular level, one of the major molecules of information transfer is what we call as the deoxyribonucleic acid, or abbreviated shortly as the DNA. And the DNA is made up of nucleotides. What you see here is a nucleotide, something like this, has a phosphate, has a 5-membered sugar, and has a base, something like this. And if you go in the direction, what I've written here, 5 prime, 3 prime, 5 prime, 3 prime, 5 prime, 3 prime, what we notice is that the bases can be of different kinds. And in nature, there are essentially 4 kinds of bases, what I've written here as adenine, which essentially partners with thymine, and I will show you how it partners. So adenine will necessarily partner with the thymine, and cytosine will essentially partners with the guanosine on the other side. So there's a sequence that is emerging as we see here, going from 5 prime to 3 prime direction in the particular direction, vertically below. What you see is a sequence that is appearing as T, G, C, A. And if you were to look at the partners, you would have T, G, C, A on this direction as well. And DNA makes use of this in a very, very interesting manner. DNA indeed forms a double strand. 2 strands of DNA are formed like this, what you see in this particular figure. And both the strands are running anti-parallel to each other. One DNA strand from 5 prime to 3 prime is running from top to bottom like this. Other DNA strands in the 5 prime to 3 prime is running from bottom to top like this. And what you notice in the center, if there is an adenine here, there is a thymine here. And if there is cytosine here, there is a guanine here. So the bases now appear in a particular sequence. A, T, G, C, G, C, A, T, G, G, C, A, P, T. And exactly complementary sequence would be there on the complementary strand. That is the way information would be transferred from generation to generation. How? For example, our mother and father had this particular DNA, and if this DNA were to replicate or make a copy of itself, the 2 strands would separate out like this, what I've shown you here in blue. And a new strands would be synthesized in the cell, what I've shown here in the green. And the way the new strand is synthesized, it would reproduce the sequence of the parent strand identical in a complementary manner. On the parent, there is a G. On the daughter strand, there is a C. On the parent, there is a T. On the daughter strand, there is an A. Bingo. We have found an exact mechanism of replication of information transfer from generations to generations. Exactly the same sequence as occurs here is now copied onto 2 molecules of DNA. And that is how information is transferred from generations to generations. Although the information is transferred from generation to generation through the molecule of DNA, cells have many other functions to do. As I said, cells have to metabolize, cells have to generate energy, cells have to grow, cells have to divide. And all these functions of the cells are what we can call as housekeeping functions. They are carried out by molecules called as proteins. And proteins also are polymers. Just like what we saw DNA, a linear polymer made up essentially of 4 different nucleotide kinds. Proteins also are polymers. And proteins also are made up of a very simple molecule, what we call as an amino acid. An amino acid because it is an amino group and it has a carboxylic group, an acid group. And is a tetrahedral carbon and has one particular group here, which can be of 20 different kinds. What we saw in DNA, there are 4 kinds of bases. In amino acids, there can be 20 different kinds in nature. So the DNA genetic code essentially specifies for 20 different amino acids here. And the amino acids then join head to tail fashion and form a linear polymer just like the DNA did. So if you have one amino acid here and has an R group called R1, it joins with another amino acid with R2 and yet another amino acid R3 through what we call as the peptide bonds here. So once again, it's a linear polypeptide that forms an protein. And to give an example is that of insulin. All of you have heard of insulin. Insulin is a very small peptide and has number of amino acids joined back to head to tail in a fashion like this. The first amino acid is glycine, the second one is isoleucine, the third one is valine, and the fourth one is cysteine. And this is the insulin chain what we see here, which is a protein. So proteins essentially are linear polymers of amino acids joined together by peptide bonds. And each protein has a specific function to perform. There are many different kinds of proteins in our cells, in our body, and all of them have different kinds of functions to perform. I've shown you here some examples. This is a very complex assembly of different polypeptide chains. This particular complex assembly generates ATP in our body. When the sugars such as glucose or fructose are broken down, the eventual molecule of energy ATP that has to be generated. And this is an enzyme which generates ATP in our body. Look at how complex it is. In fact, it is also embedded within the cell membrane. What you see here is a cell membrane which is made up of lipids, as we talked a while ago. And there is a gradient of protons going in one direction. And a gradient of protons, when it goes, it rotates this, what you see here in green, blue, and red. And once it rotates, the rotation is transferred by a shaft to the enzymatic subunit, and they also rotate. Is an example, a real life example of a molecular motor. And during rotation, an ATP is formed at this particular active site of this particular enzyme. That's one example of proteins and the function that they do. Another example of a protein is offered by this particular protein. Is a deadly protein called cholera toxin. This protein is used by the bacterium which causes cholera. A bacterium called Vibrio cholerae. Vibrio cholerae secretes cholera toxin into our guts, into our intestines. The cholera toxin binds on the epithelial cell surface, our intestinal cell surface, through this 5 pentamer of these subunits. And the moment it binds there, it facilitates entry of this blue or the cyan colored protein inside our cells. And that triggers a deadly set of reactions which eventually lead to loss of sodium and potassium ions from our cells and water. And that's what we say is what we have diarrhea or dysentery. And that's how cholera onset happens. But this cholera toxin, what you see here, is also a protein. And it helps the bacterium to enter the cells and break cells open. We see another example here. These are antibodies. Many of you have heard of antibodies in recent times, right? During the COVID pandemic, many people asked whether you have antibodies against COVID or not. Antibodies are our body's natural defense against invading organisms. Antibodies binds to invading organisms, what we call as antigens, and destroy them. So the job of our immune system is essentially to recognize if any invading organism is entering our body, and if it is, recognize that and destroy that. And one of the major parts of that done is by these particular antibodies. These are also proteins. Another example of proteins are the proteins called histone proteins, what you see here in the center. I told you, DNA is a molecule of information. And we have in our body roughly about 1 to 10 trillion cells. 10 to the power 12 or 10 to the power 13 cells that make up our body. Each cell carries a DNA in its nucleus. If you take one DNA molecule from one cell and extend it like a linear polypeptide, the length of that would be of the order of about 1 meter. Believe me, one DNA molecule from each cell of our body would be 1 meter long. Our cells are not 1 meter long. Our cells are 1,000 times smaller than that. How does body compact this DNA and compact that into one single cell? And that is the job done by these histone proteins. The DNA simply wraps around these proteins. And wrapping around offers a very effective mechanism of compacting the DNA. Now you see that a string which would have been 1 meter long can be compacted 1,000 times or even more than 1,000 times so that the DNA can be accommodated in each of the cells. These histones are proteins. What I'm trying to give you is a flavor of functions of different proteins in the cells. So although DNA is the molecule which carries information, all the functions are done by proteins. And one example that we'll talk a little bit more is that of hemoglobin. All of you know that hemoglobin has a function. It has to carry oxygen from our lungs to different parts of the body. It has to carry carbon dioxide from different parts of the body into the lungs and release into the air. That's exactly what is happening when we are breathing in and breathing out. When we breathe in air, oxygen gets enriched in our lungs. Hemoglobin binds to that oxygen. Simultaneously, heart is pumping up. Remember, heart is pumping. And the pumping of the heart allows the blood to be transported from different organs to another organs. For example, from lungs to our fingers. And while doing that, hemoglobin travels in this blood, carries oxygen from the lungs to our fingers, and releases oxygen in these fingers. That's exactly what hemoglobin does. And hemoglobin is a major molecule of our red blood cells, or what we call as RBCs. What you see here, platelet structures, are the RBCs. Of course, this is not to the scale. Millions of hemoglobin molecules are accommodated in one single RBC here. What you also see here in the figure are also RBCs, but from those unfortunate people who have sickle cell anemia. So those people who suffer from sickle cell anemia, their RBCs don't look disc-like like this, but rather like sickles here. And that happens because of a single mutation in hemoglobin. So when I told you that information is passed from generation to generation, there can be error in that information. When the DNA is replicating to form two daughter copies, there can be an error. And such errors, what we call as mutations. And if there are mutations in our DNA, for example, one particular amino acid here, what we call as a glutamate, at the 6th position in the hemoglobin, if it becomes a valine, that mutation can be so deadly that can change the shape of RBCs from disc-like to sickle cell-like. And let us look at this particular video, how that happens. Doesn't matter. The video is not playing, but what happens is that because of the water-hating nature of valine and water-loving nature of glutamic acid, in the mutation when there is valine, it develops a patch, what we call as the hydrophobic patch. Hydrophobic patch is the one which does not like water. And therefore, different molecules of hemoglobin, 100 million copies of hemoglobin molecules can aggregate through the patch which does not like water, what we call as hydrophobic. And when 100 million molecules of hemoglobin cluster together, they force a change in the shape of the cell into this kind of a cell. And that's actually what causes sickle cell anemia. So there can be mutations, and many such kind of mutations can be deleterious. Many mutations can also be useful. And we have seen that many mutations can cause different kinds of diseases in nature. And that's a very fascinating example of how mutations in proteins can cause diseases. So what we have seen till now, we have seen what are the building blocks of living molecules, living systems. We started with atoms, carbon, hydrogen, nitrogen, oxygen, sulfur, and phosphorus. Then we said that all living forms have very similar kind of molecules. They go on making more complex molecules, assemblies, and larger molecules. All the molecules together assemble to form cells in our bodies. And I told you, 100 trillion cells in our body. The cells together form complex organisms like ourselves, human beings, animals, plants. We are all very complex organisms. And that makes living systems in the end. So what we have seen is a journey starting from remarkable unity in diversity of molecules all the way to many different living systems that exist on the planet today. The life is so diverse, but yet it has beautifully used the same kind of architecture of atoms and molecules to make up itself. Very fascinating. I hope you would agree with me on the fascinating aspects of this. So the complex assembly of molecules, cells, and organs can also be summarized in one short form. Our body is made up of many different kind of cell types. Right? We have blood cells, we have pancreatic cells, we have muscle cells, we have heart cells, we have brain cells, we have lung cells, and so on and so forth. And in number, there could be many. Brain cells, for example, could be 10 to the power 11 cells in our brain. They don't get recycled generally in their lifetimes. But blood cells, about 10 to the power 10 blood cells in our body, and they do get recycled every now and then. Every few days, the entire blood cells would get recycled. There are bone cells, the liver cells, and the body, by doing so, using all these cells and the molecules within them, has made sure that it can sustain itself. If there is a damage on our skin, it can be immediately repaired. If there is a damage on liver, it can be repaired. If there is an external organism which comes and invades our body, the body has a beautiful immune system that can take care of it. But yet, there are diseases which we still have not found answers. And our endeavor is to find answers to many such interesting questions. A large number of questions that come to our mind, I've just highlighted some of them. And the generation of the students who have joined me today on this virtual webinar, I urge all of you to take up these questions and make sure that we advance our understanding of nature for future. How did life begin? What makes us humans? We have heard of consciousness. We know about consciousness, but we don't know what is consciousness. What is consciousness? We are surrounded by so many different life forms around us. Bacteria, viruses, other organisms. How have we learned to live with all these different life forms around us? We get diseases by either genetic mutations or by invading organisms. Can we cure all diseases? Diabetes, cancer, Alzheimer's, tuberculosis, malaria, so on and so forth. Can we find answers to all the diseases? Can we live forever? I'm going to leave with these questions with all of you. Answers to any of these questions are not known, and it's up to you to take up science and try to answer some of these questions. Thank you all very much for joining me here. And if you have any questions, I'll be very happy to answer them on email. But Shubda, if there are any questions, I would also be happy to answer here for the next 10 minutes or so.
Yeah. So there's one question, sir. Is a cell in bacteria and a cell in human same?
So we cannot call it same. They are similar. The lipid around the bacterial cell and human cell, we can say they're of similar nature. They're not identical nature, but similar nature. The cytoplasm that is within the cells, that also is of similar nature. Bacteria don't have nucleus, so the DNA is freely floating around in cytoplasm. Well, our body has a nucleus, and the DNA is compacted within the nucleus. So there are similarities, and there are very major differences. Is that what we have to understand?
Okay, thank you, sir. And there is one more question that is, how do genes control the growth and division of cell?
So genes are naturally programmed for this, and the questions of such kinds are not very well, answers of them are not very well known. But nonetheless, what we know is that genes have programs for cell development, growth, and eventual death. So because of this program, the cells can grow, the body can grow, the organs can grow up to a particular extent. It cannot grow infinitely. And in fact, if we lose control on the growth of that, it causes what we know as diseases such as cancer and so on and so forth. So genes are programmed to do this. We don't know answers of many of those, and are being hunted as we speak today.
Okay, thank you, sir. Thank you for the lovely talk. And as you have just rightly said, there is a remarkable unity in diversity in all life forms, and right from the building blocks to living cells. And yes, there are many events that occur in the life of a cell. And as you explained, different functions of protein, the building blocks of life. Are very much benefited. I think students must be very much benefited by your lecture. And thank you so much, sir. Thank you so much for joining for this webinar series. Thank you. Thanks a lot.